the advancements in technologies have given us a look at God's creation that no one ever saw before. And so it's a remarkable time to live at the time that where we do, where there are technologies that have been developed that allow us to see aspects of God's creation that no one ever saw before. And the cell is one of those, giving us a better appreciation by looking at the incredible design within cells, giving us a better appreciation of who God is and what it is that he actually made for us. And that we've got to remember that everything that exists was made by God and made by God for us. It's incredible. Microscopic world is a fantastic place. I've spent lots and lots and lots of time on the microscope. I mean, I add up all the minutes, felt like I spent a year of my life on a microscope. When I was at Texas Tech, I don't know if you can recognize me in that picture because that's before I turned into Shrek a little bit. But when I was at Texas Tech, I worked at a lab at Texas Tech before I started graduate school that was attempting to grow cotton cells to lengths that would allow them to be used by textile industries. And as one of my jobs there, they bought a new image analysis system. This was more publicity photo, kind of highlighting some of the technologies that were available to the department at that point in time. But we bought a computerized image analysis system where I would have to analyze these cell cultures, the links in particular that these cells were getting to using this system. To my knowledge, I was the only one that ever used it. It was a $50,000 image analysis system, and to my knowledge, I was the only one that ever used it. After I was left, chances are I was never used again. That's an assumption, but that's sometimes how grant money is spent, They got to spend it before it expires, and they'll buy some piece of technology that may never get used again. I'm sure the microscope was used, but I'm not sure any of the rest of that was used. But the microscopic world can be an amazing place. It really is an amazing thing to get under a microscope. It takes a little bit to get comfortable in that environment, to comfortable moving, being able to move around in an articulate manner. But there is a beauty and a symmetry to the microscopic world that really is like nothing else we've ever experienced. I kind of describe it as almost, if you've ever been scuba diving, scuba diving is very foreign to our senses. And in one way, the microscopic world is similar in that regard. It's so foreign to our experiences that it is like entering, an alien world. There's a beauty and a symmetry to it all that is just captivating. So, so beautiful. But we also know that within this microbial world, exist many of the organisms that are responsible for the diseases of old or those of modern day. Diseases like leprosy in the Bible we now know was caused by a bacteria, or ulcers. Up until recently, they didn't know what caused ulcers, and the only remedy for them was to take antacids. And I don't know if you have noticed, but think about how long it's been. Do you see antacid commercials anymore? I mean, back in the day, a few decades ago, their antacid commercials were on the TV all the time. And I bet you the old timers could, like at my age, could think of several different brand names of antacids. But you almost don't see them anymore advertised in any significant way because eventually they learned that ulcers were caused by a bacteria that was resistant to the acid that's in the stomach. And now all you have to do is take a course of antibiotics and you're cured of ulcers. But until they developed these antibiotics, until they had diagnosed it accurately and developed the antibiotics, there was no cure. All you could do was take antacids to stop it from burning. And they would tell you that you would get ulcers, you could give yourself ulcers through stress. They would tell you, don't stress yourself out, you can give yourself an ulcer. We found out it was a bacteria that was resistant to that acid. This specific, the red cells in this image are a bacteria, and this specific one is the bacteria that causes food poisoning. So if you eat improperly cooked pork or even eggs, you can get food poisoning from this bacteria called Salmonella. Bacteria serve many they are beneficial, extremely important. It is estimated that perhaps close to 40% of the cells that make up the human body that you're walking around with are in fact bacterial cells. Close to 40%. Right? Estimated 100 trillion cells that make up the human body, but 40% of that total number, in fact, are bacterial cells. They help you digest your food. Your colon or large intestine are just full of bacteria that are part of your digestive system, an integral part of your digestive system. They are very important, but in the ecosystem, they serve as decomposers. Bacteria and fungi are the decomposers. You need producers in an ecosystem, the photosynthetic plants. You need consumers in an ecosystem, and you need the decomposers so that all of that dead and decaying matter is quickly decomposed and made available, again, to the next generation. So they're very important, but if you get a bacterial cell, in a cut or, somewhere, it'll start trying to decompose you before you're actually dead, and that's why they can be very problematic. And specifically because they grow and divide so rapidly. This is a time-lapse video, but bacterial cells can grow and divide about once every 20 minutes under optimum conditions. So that's incredibly fast rate of reproduction. And at that rate, if you get one in a cut, they can cause some serious problems. Basically trying to eat you is what they're trying to do. And some of them have caused real problems. I mean, the bubonic plague was caused by a bacteria. Bubonic plague was. But it is a beautiful thing. Just spending some time under a microscope, just a beautiful thing. And suffice it to say, we now know that these microbial organisms, like the ones you see here, these are all single-cell organisms, that these cells like this are everywhere. Any little puddle of water that you see out there, where there's some dirt, if there's some dirt and some water, in that dirt is decaying matter, in that environment, you're going to find cells like this. So when you walk, think about all the times you walked around in bare feet through, standing water out there, or a little bit of mud or something. That's the kind of stuff crawling up between your toes, just go to tell you. These single-cell organisms are everywhere. But one of our main focus for looking at cells tonight is to look at them for the evidence of design that's there, to point out some of the interesting design features that we find in cells. But it kind of begs the question, how do you know if something is designed? Can you tell? Because the big debate here, of course, is that none of this was designed. It all just developed all on its own. So how do you know if something's designed? I mean, I argue that observation alone can tell you for many things. If you were to see, if you see Mount Rushmore, if you go to Mount Rushmore and you see those sculptures, can you tell just from observation that those had been made rather than having been eroded to those shapes naturally? Or when you're walking along the beach, if you see a stone on the beach and you pick that up, could you tell that stone had been shaped by human hands, like into an arrowhead? Can you tell just by observation that something has been designed? For many things, I say that you would say yes. And today, we're able to look at cells under magnifications that allow us, I think, to say conclusively, just from crude observations alone, that what we're looking at there was made. One of the most powerful microscopes in existence are called the electron microscopes. There are optical microscopes that are used in classrooms, around the world that only get you up to about 40 power magnification. 100 power if you use the oil immersion objective, but standardly you get to only about 400 power magnification. Scanning electron microscopes can get up into the tens of thousands of magnification. I'll show you a few pictures of these from what's called a scanning electron microscope. And again, just to show you some cells. This is one cell of a colonial organism, but I argue that if you saw that, if you're walking along the beach or somewhere out and you saw that laying on the ground outside, would you assume that had been made or that was the result of purely natural processes? Or what about these? If you're walking along the beach and you found one of those lying on a beach somewhere, would you assume that had been made rather than having formed through purely natural processes? Now let me give you an idea of the scale that we're looking at here. You see the little bar on the bottom left. That says that's the size of 20 micrometers. Okay, the little U sign, backwards U sign, is a sign for a micron. Okay, in this case it means micrometer, which is a millionth of a meter. Okay, so bring this down to a scale you can envision. A centimeter is about the width of a dime. Okay, and on a centimeter there are 10 millimeters. 10 millimeters in a centimeter, and there are 1,000 microns in a millimeter. The human eye can see a millimeter, and it can even see a half a millimeter. But what you're looking at there are 20 microns, and again, there are 1,000 microns in a millimeter. So you're talking about something incredibly small, incredibly small. Or look at this one. Again, if you saw that anywhere else, just because it's small doesn't mean that our observations are any less true. I think anyone, if you saw that laying on the ground or anywhere, that you would just, I think we can say that that was design. I'm going to show you this. This is a single cell alga for singular. This is a single cell alga that is making armored plates for itself. You saw just for a second there as the technician was panning up and down the plates that this thing is covering itself with. This is called a coccolithophore, and it's making little calcium carbonate plates to armor itself that are called coccoliths. But this is a standard optical microscope at about 400 power, and I just want to show you what it looks like under an optical microscope so you can better appreciate what it looks like under a scanning electron microscope. Look at this. I mean, I think just images of these cells from these high-power microscopes will tell us, yes, that definitely looks like something that was made. And of course, the Bible tells us without question that God made everything. For by him all things were created, in heaven and on earth, visible or invisible, whether thrones or dominions or rulers or authorities, all things were created through him and for him. The Bible is clear that God made everything. Everything that exists, all matter and all energy in what we call the universe was created by God, and all of that was created just for us. But of course, we live at a time where there are many today who teach that everything we see is the result of purely natural processes. Naturalism is the reigning philosophy of the scientific community. The vast majority of practicing scientists today argue that natural processes alone were responsible for everything. That everything we see came into existence through purely natural processes. This is what we call naturalism. They would argue that all physical phenomena must be explained as being the result of purely natural processes. The current position of the scientific community would argue that there are no such things as miracles, no such things as supernatural events. There was no creation. This view is diametrically opposed to biblical creation. And unfortunately, a lot of Christians just don't understand this. They don't understand that evolution or Darwinism is in direct opposition to biblical Christianity. The Darwinists recognize this. Your atheists, your Darwinists understand this. That's why they use the Darwin fish. Have you guys seen the Darwin fish that people put on like the backs of their cars, you know, a little decal on the backs of their cars? What are they saying by doing that? They're taking the Jesus fish. Everyone understands what people use the fish. It's been an ancient symbol of Christianity all the way back to the 1st and 2nd century, literally, because Jesus' first few disciples were fishermen. And they even found fish motifs on the house that is attributed to Peter's house in Capernaum, where it's probable that Jesus lived for a period of time. They've excavated what they believe was and it was probably the house where Jesus had lived, today described as Peter's house. But anyway, they found even fish motifs there in the walls of the home where probably Jesus had lived while he was in Capernaum. But they've taken the Jesus fish, this ancient symbol of Christianity, stuck a couple of legs on it and say, no, I reject your truth of creation and Jesus, that all things were created by him and for him. I reject that, and I insert my own truth that life came about through Darwinian mechanisms, not creation. That's what the use of the Jesus fish in that context is. They're saying, nope, I reject your truth about God and Jesus, and I argue that evolution alone is responsible for this. But what we need to keep in mind, as far as the evolution is concerned and the origin of this thinking, is that at the time Darwin and his contemporaries were devising these theories about evolution, they knew almost nothing of the cell. They knew almost nothing of the cell. The first person to actually see a cell, as far as we know, did so in 1655. At least the first person to describe seeing cells was this guy by the name of Robert Hooke, who was looking at a thin cross-section of cork and saw what looked like little compartments. Of course, we now know that those are the cell walls, And he said that they reminded him of the cells that he has seen in prisons or monasteries. So the little living quarters, we call those cells. He said they look like little living quarters, like cells. So he coined the term. But the cell theory, the theory that organisms are made entirely of cells, that all life processes are performed by cells, this didn't come about until the early to mid 1800s. The cell theory, the knowledge that organisms are made entirely of cells or cell products, and that all cells come from pre-existing cells. This theory, this knowledge of the cell itself, did not come about until about the time Darwin and his contemporaries were forming their theories about evolution. The Origin of Species was published in 1859, yet the cell theory didn't form until between 1837 and 1855. They knew almost nothing of the cell at the time Darwin published Origin of Species, almost nothing. In fact, Ernst Haeckel, who arguably is one of the most famous evolutionists of Darwin's day, a bit infamous today. Most people will know Ernst Haeckel for an act of fraud that he committed in an attempt to prove evolution called Haeckel's embryos. He commissioned someone to construct drawings of embryos at various stages, and his point was that, look, if you look at the embryos of all these various animals, they look identical at the same stage, pointing to a common ancestry. He argued that, look, you can see, but now we know that the embryos look nothing like that at those stages, and that was recent. This was only recently, within the last 20 years. Did they republish Haeckel's embryos in Scientific American and took him to task for that? But Ernst Haeckel named the bacterial kingdom. So he's a biologist, a cell biologist, an embryologist in his own right, to the point where he named the bacterial kingdom. He named it the kingdom Monera, which is what I knew it as when I was studying biology. They've taken the bacterial kingdom and split it into two kingdoms, now called Archaebacteria and Eubacteria, but he was the one who originally named the bacterial kingdom. And this is how he described the bacteria. This is Ernst Haeckel. And look at the date on this, 1883. Again, Darwin published Order of Species in 1859. So this is how they viewed the cell, even 25 years or so after Darwin published Origin of Species. This was the view of the cell. He says, the cell is not composed of any organs at all. Speaking here specifically the bacterial cell, not composed of any organs at all, but consists entirely of shapeless, simple, homogeneous matter. It's nothing more than a shapeless, mobile little lump of mucus or slime, consisting of albuminous combinations of carbon. That was the view of the cell in Darwin's day. It's nothing more than a little lump of mucus or slime. That's it. I mean, when that's your view of the cell, it's easy to envision a cell forming all on its own through purely natural processes in some warm little pond, as Darwin described. But now we know the cell is vastly more complex, and the bacterial cell as well. The bacterial cell, in fact, has one of the most complicated molecular machines we know of, called the flagellum. I'll show you the flagellum a little bit later. But it's far from a shapeless little lump of mucus or slime. Cells do have organs. He said they don't have organs. We know they have organs. We call them organelles because they're little mini organs. They have organs. In comparison to our marvels of modern technology, cells have orders of magnitude more moving parts. Tremendously complex is the cell. And again, they do have organs. We call them organelles. We'll talk about some of these. The mitochondria, I'll show you the mitochondria, is some people call it the powerhouse of the cell. Another organelle is the chloroplast, not shown here, which is involved with photosynthesis. The Golgi apparatus, or shown up there, is kind of like a chemical factory for the cell. But it does have these little organs, we call them organelles. And cells come in vastly complex forms. The human body, for example, has cells that are in numerous highly specialized types. For example, the muscle cell or muscle fiber you see there in the upper right-hand corner is a very specialized cell that performs the function of contraction. It shortens. And in shortening, it performs tasks like moving bones so that we have movement, squeezing the blood that's in the heart to perform circulation. Muscles, but it's tremendously highly complex and specialized cell. The hair cell that you see there are the cells that are in the inner ear that are involved with both hearing in the cochlea and balance in the semicircular canals. A very specialized sensory cell. The nerve cells are involved with carrying signals from one part of an organism to another. They send messages through an electrical signal. And they're the longest cell in any organism. In humans, you have nerves, a single nerve cell that is about 3 feet in length. The ones that connect to your spinal cord and run down and control the muscles in your calf are about 3 feet in length. In a giraffe, you have single nerve cells that run their neck that are closer to 9 feet in length. One cell, 9 feet long. The largest cell, though, by volume is the egg in any organism. The egg, prior to fertilization, is one cell. So an ostrich egg, one cell. That's a big cell by volume. Cone cell, the one on the far left, is one of the cells in the eye that's responsible for vision, for detecting photons of light. So you got these vastly different complex cells in multicellular organisms. And again, in the human body, there's close to 100 trillion cells. The human body makes or replaces well more than 2 million cells per second. You use and replace more than 2 million cells per second. That 2 million is actually just the number of red blood cells that you use up and make every second. So that's a lot of cells. Now let me read you through how an evolutionist describes the cell now. Remember, back in Darwin's day, it was described as a simple little lump of mucus or slime. Listen to how an evolutionist of our day has described the cell. This is Michael Denton. He says, to grasp the reality of life, as it has been revealed by molecular biology, we must magnify a cell 1,000 million times until it is 20 kilometers in diameter and resembles a giant airship large enough to cover a great city like London or New York. What we would then see would be an object of unparalleled complexity and adaptive design. Back in Darwin's day, cell was viewed as a simple little lump of mucus or slime. Fast forward 150 years, what we now know is the cell is an object of unparalleled complexity. Nothing parallels the complexity of the cell. It is like a city. It is that complex. An airplane doesn't compare. Even an airship, or whatever he was talking about, alien spaceship. It's like a city full of complexity that's miniaturized down to a microscopic scale. That's how big the cell is. This is just an animation produced by Harvard. It was produced to highlight one specific new discovery, but an excellent little survey, a look inside the cell, this animation. But it is. The cell is like a city. It's cities worth of complexity. Highway systems, transporting things from one side to another, all kinds of factories and motors running around doing work. It's got a skeleton of sorts. What you're seeing right there are fibers that make up a skeletal system for the cell. And as well, some of these fibers serve as a highway system for transport. And it can assemble and disassemble these in a microsecond. The processes that you're showing here are greatly slowed down, but these things occur in literally a microsecond. That's one of the fibers called a microtubule. I'll talk about those a little bit. And this is a motor protein called a kinesin that's carrying a vesicle down one of these microtubules. But instead of running at a snail's pace like this, you actually have highways, masses. I'll show you a video here in a second where you'll see whole highways of these things in operation. That's the genetic code right there. I'll talk a little bit about the genetic code and what that's used for and what the cell does with that. That's one of these organelles reading the genetic code. That's the Golgi again. Golgi is like a chemical factory. But regarding the cell, today we really only understand the big picture. We understand what some things are doing, but the reality is we know very little of what's happening. Because it's like, I kind of use the analogy that our knowledge of what we're doing is in studying the cell, what we're doing is like looking at a city from a distant hilltop with a telescope. That's kind of what we're doing in our studies of the cell. Periodically, we'll squish one of them, and then we'll pick up the pieces and look at the pieces and see how they fit together to try to understand what's happening. But the reality is we can't see the cell in operation. At the current levels of magnification, we can't really see a cell at that the magnification of this specific animation. We can't see that with microscopes. Not even the most advanced microscopes. We can't see these things happening. The electron microscopes will only look at things that are dead. The highest power microscope, the one that we were looking at images from before. You can only look at things that are dead with that level of magnification. So you really can't just dive in and see these processes as they're happening. We have to look at it through a microscope for some of these. And so we know some of the big picture. We know some of the big processes, but much of what's happening in there is still a mystery. But I want to point out a few things that we do know that are just tremendously complex. And one good place to start is to look at some molecular machines. There are machines, lots of machinery in the cell that are made of molecules, and so we call these molecular machines. And these molecular machines, I'll talk about a couple at least. There's an argument for intelligent design that we could use when talking about these machines called irreducible complexity. And I know this is a big fat term, so I'll try to unpack this a little bit for you. The term was popularized recently by Michael Behe in a book that he wrote called Darwin's Black Box. But and he in this book he fleshed out the irreducible complexity argument for intelligent design and how that worked. But and to do this, he used a mousetrap because the mousetrap is the simplest machine we can think of. I mean, there's nothing really simpler as far as a machine is concerned than a little simple mousetrap like the one you're looking at right there. But what an irreducibly complex machine is one that cannot be reduced to individual components that serve a function. So something like a mousetrap only works if all the components are there and it's completely assembled. And at that point in time, only will it function. Now, what Darwinists have to explain is how these molecular machines came into existence through numerous successive slight modifications, as Darwin described it. But a machine, like a mousetrap, doesn't work unless all of the components are there. Darwinists have to explain how one of these machines came into existence by first having just one piece of it. But a mousetrap like this won't work if you just have one piece. And a cell is not going to just keep making that one piece generation after generation after generation after generation until it happens to stumble across the next piece. And then keep making those two pieces, even though they don't work, because you don't have all the pieces yet. It's not going to keep making the next piece over and over and over again. These machines only work unless all the pieces are there at the exact same time, and it's assembled. So he describes it like this. He says, an irreducibly complex system, he says, this is Michael Behe, is a single system which is composed of several interacting parts that contribute to the basic function, and where the removal of any one of these parts causes the system to effectively cease functioning. An irreducibly complex system, he continues, cannot be produced gradually by slight successive modifications of a precursor system, since any precursor to an irreducibly complex system is by definition non-functional. Since natural selection, he continues, requires a function to select, an irreducibly complex biological system, if there is such a thing, would have to arise as an integrated unit for natural selection to have anything to act on. It is almost universally conceded, he says, that such a sudden event would be irreconcilable with the gradualism Darwin envisioned. So these these molecular machines are just far too complex for Darwinian mechanisms to explain them. And Darwin himself admits to such a thing. This is Darwin from The Origin of Species, 6th edition. He says, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. But I can find out no such case. Yet today, with our knowledge of the cell and these molecular machines, we can point to many examples of things that are just simply too complex to be formed by Darwinian mechanisms of numerous successive slight modifications. We can point to many. Let me show you a few of these. Machines are very important in cells for things like movement. Cells have to move, and they have to move a lot. Not only do some cells have to swim around, but they also have to transport things. I want to look at this video at the highway systems I was talking about. Remember that little kinesin protein walking that one little vesicle down that microtubule? You got highway systems moving from one side of the cell to the other as they transport things. Or look at this one. Now, this one is time-lapsed, but it'll give you a sense of just how much work is taking place in a cell. Watch this. Now, this is a time-lapse sequence. It's sped up. This whole thing actually occurs over the course of an hour. But look at all that's taking place there. It's a lot of work, a lot of processes going on. This is actually a cell preparing for division. And you can see it right in this nuclear membrane breaking down right there. The chromosomes are now being moved around and now pulled apart. So that's a cell that was in the process of getting ready for division. But when you see an image like this, then you start to get the sense of what I'm talking about. That what you're looking at is a city full of activity, a city full of work taking place, and a city full of complexity, all right there. Cells have to move. They have to move. All of the parts that are there have to be moving and active, just like all of the machinery in our cities have to be moved from one place to another. And once you get into location, all the parts have to move. Lots of movement. All of this movement requires these machines. Cells themselves have to move. This is an amoeba, an amoeboid type cell that actually is able, this cell is able to just move wherever it wants to go. It just oozes off in any direction it wants to travel in. But to do that, it has to move everything in the cell. Remember, all of those organelles, all of that highway system, all of that has to be moved. So this is like a city. Again, think about this as a city. You got a city like Seattle that just decides it wants to up and move over to the other side of the mountains to enjoy some sunshine for a little bit. You know, it can just move everything. All of those highways, all of those factories, it can just pick up and move the whole thing. Okay, so I'm going to show you one molecular machine that's very important, and it's important to provide all of the energy that's needed. And the energy in a cell is provided by this molecule. Now, I'm going to go in a little deep here for you, but I hope you can appreciate a little bit extra detail in your understanding of how cells work. This is the molecule that provides energy for everything that has to be done in a cell. And we use energy for lots of things, like muscle contraction. To move muscles, it uses this molecule called ATP. Nerve impulses use this molecule called ATP. Everything that requires energy to do work requires ATP, everything, okay? And so you probably know that this is a model of a molecule, okay? If you didn't, all of those letters that you're seeing up there, the O's and the P's and the N's, those stand for elements. So the N stands for nitrogen, the O stands for oxygen. The carbons in a model like this, or just a diagram like this, are just not labeled, because this is an organic molecule that contains lots of carbon, and it would be excessively redundant to label all those carbons, nor are the hydrogens labeled. So it's kind of abbreviated diagram. But has a bunch of carbons in there too, and a bunch of hydrogens, just none of those are labeled. But it does, so it's what's called a triphosphate. ATP stands for adenosine triphosphate, and tri means three. These are the phosphates right here on this little arm to the right. Each of those little PO3, PO4 molecules, those are phosphates. It has three right now, but these atoms, these elements are held together by bonds, and if you break one of those bonds, you can transfer the energy to do something else. And that's what it does. It cleaves off one of those when energy is needed, one of these organelles or one of these molecular machines like that transport protein, that kinesin, it'll cut off one of those phosphates. The energy from that phosphate will be used to do some work, and it changes into adenosine diphosphate. Okay, that's how energy is transferred from ATP. I'll get a little more information than you perhaps wanted, but now it has to remake it. For you to have more energy, you have to make another ATP. Your body uses and remakes half of your body weight in ATP every day. That's how important ATP is. All of the chemical reactions in your body, all of the processes in your body that need energy, get it from ATP, and so you have to use half your body you use and remake half your body weight in ATP every day. Poisons, the cyanide is a particularly nasty poison, cyanide, and the reason why it's so deadly is cyanide blocks ATP synthesis, and in doing so will kill you in 30 seconds. So if you can't make ATP, dead in 30 seconds. That's basically the end story. Well, the organelle that makes ATP is called the mitochondria. Some call it the powerhouse of the cell, because what it does is it makes energy molecules, energy-carrying molecules. It makes these ATPs, and it makes them from glucose, okay? Plants make glucose in photosynthesis. We eat plants because that glucose molecule is an energy carrier. Lots of energy in glucose, okay? But it's more energy than your body needs to do work. And so what this organelle is doing is basically, it's like converting a big energy carrier to a small energy carrier. Or you can look at it as like it's converting a $100 bill into a whole bunch of quarters. Because for the work that you need to do, all you need is quarters. But the glucose molecule is a big $100 bill, and this converts it into a bunch of ATPs. You can think of it like quarters, okay? Mitochondria. Let me show you, though. So we're going to have, we're going to take a little quiz at the end, so make sure you pay attention. You're going to need to know some of this stuff. Okay, this is part of the chemical reaction that's necessary to convert glucose into ATP. And this is just one example of many chemical reactions that are taking place in the body, and I want to impress upon you how complex this all is, what is necessary for just one chemical reaction to convert glucose into ATP. How much is required just for this one thing? Okay, this diagram is showing some of the chemical reactions that are necessary to convert glucose into ATP. This part of it's called the citric acid cycle. Okay, now it doesn't show it, but at the very top of the screen, just off screen, is the glucose molecule. The first step is it's converted into pyruvate, which you see at the top of the screen. Next, pyruvate is converted by an enzymatic reaction to acetyl-CoA. Then acetyl-CoA is combined with oxaloacetate, and you make citrate. Okay, then citrate is converted into isocitrate by an enzyme shown there in pink called iconitase. Isocitrate is then converted into alpha-ketoglutarate with this enzyme right here called isocitrate dehydrogenase. Then alpha-ketoglutarate is converted into succinyl-CoA. Succinyl-CoA is converted into succinate, succinate into fumarate, and each of these steps requires a new enzyme. An enzyme is a protein that performs a chemical reaction. And so you need all of these enzymes: isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, succinyl-CoA synthase, succinate dehydrogenase, fumarase, malate dehydrogenase. You need all of those enzymes. All of those enzymes have to be there to just perform each step of this process that's required to convert glucose into ATP. But what you're really making during these reactions is this molecule, where is it? Right here, NADH. You see that? NADH plus the hydrogen ion H plus. It's making those. One here, and one here, because those are used to power this, which is the ATP synthase. And these are in mitochondria in I don't remember, hundreds of thousands as far as I remember. Whole bunch of these in a single mitochondria. This is a little molecular machine. Now, this was discovered, I don't have the actual discovery date, do I remember? But the guy who discovered this won the Nobel Prize in 1997, okay, for discovering the ATP synthase. And what this does, it uses those NADHs and those hydrogen ions to spin this. Hydrogen ions flow through it, and the top of this thing spins at 200 times per second, and every time it spins, it makes 3 ATPs. And it spins because hydrogen ions flow down through this thing. But it's a rotary engine. It's a complex molecular machine that's found there in the mitochondria. And it is composed of 500 individual proteins. This is just a diagram, a three-dimensional diagram to show you all of the individual proteins. Every little dot that you see on that diagram right there is a separate protein that is made. And there are 500 individual protein subunits just used to make this. Okay, so what do you need to make ATP? Okay, you need all of those enzymes that I was talking about to break down glucose, make those NADHs, and then you need the NADHs, and you need the ATP synthase up there. One other thing you also need to make ATP, you need ATP. You need all of those enzymes. You need the ATP synthase in the mitochondria, and you also have to have ATP to get the whole thing started. But this seems to be common in in the world of the cell, the chicken or the egg argument. How do you get this without this, and which of these came first? Huge problem. I'll give you a couple more examples of that. And one more molecular machine I want to show you, one more important molecular machine is the cilia. This is a ciliate, a single cell organism that swims through water because it's covered in little microscopic hairs called cilia. So just watch this for a little bit. You see it's covered in cilia. It also has some around its mouth. Did you see it eat something right there? It's got some cilia right around a little mouth pore that it has, and it can channel water. It can channel stuff into its mouth, direct stuff into its mouth to help it eat little bits of debris or algae, those kind of things. Cilia, I'll show you another one. This is another single cell organism that's covered in cilia, so we call it a ciliate. But this is an organism that lives in the guts of termites. So you want to know why your termite is so effective in eating your house? It's because you got a bunch of these things in its stomach helping. But even that's a single cell organism, but it even has a head. Did you see it? But it's a single cell organism. Incredible. And all of those things that you're receiving, that waving action, of course, that was the cilia. The cilia. And cilia are found throughout, the cell and animal kingdom. The single cell organism cells, like we just saw, those ciliates, use them to swim through water, through aqueous solutions. You also see them in a lot of other animals. Frogs have them on their tongue, so they can, using cilia, it helps them move things to the back of their mouth with from their tongue. But these are actually lung cilia. You have cilia in your lungs, covering the respiratory passage in your lungs. You have cells that'll secrete mucus, and then they have the cilia that move the mucus up to your esophagus, and once that mucus and the debris that got caught in the mucus, you swallow that, So in a way, now there are other animals that do it, something similar, like clams. Also, they will draw water in through a little pore. They draw water in using cilia to help draw this water in. The body cavity will be covered with mucus, and the cilia they collect debris that they breathe in with the water, and then that cilia will help take the debris to their mouth where they can swallow it. So clams use these cilia to filter feed. But in a way, you're doing something similar. Not to gross you out a little bit here, or actually intending to gross you out here a little bit. But, so this lung cilia is used you catch all the debris that we breathe in on this mucus, and then the cilia moves that mucus up to your esophagus where you eventually swallow it. And you know that most of the debris in the air in your household, all those dust particles, most of that are dead skin cells. So when you breathe in that air and all those dead skin cells get stuck to the mucus, and then your cilia help you swallow that, in one way you're filter feeding, but you're not filter feeding debris that's in the water you're swimming around in, but on all of your housemates there, your family members, you're kind of filter feeding on all their dead skin cells. Awesome. It's awesome stuff. So let me show you a little bit, give you a little more detail here on the cilia. This is a cross section of a cilia. They have so if you slice through the cilia and then look down through it, this is what you would see. And it is made up of microtubules. Now, on the video before, I showed you a microtubule. That was what that kinesin was walking down. That was a microtubule. They're used for many things. They're also used to pull the chromosomes apart during cell division. But here, and here, they're used as the main core, the core molecule for the cilia. Also flagellum. I'll show you flagellum in a little bit. But those little circles that you see there, those are microtubules. So it has a pair of them in the center, and then 9 pairs of them going around the outer edge. Or look at this three-dimensional diagram from the National Library of Medicine. Gives you a look at the mechanical parts and how these machine parts fit together. I mean, it is an engineering diagram that you're looking at there, because the cilia was engineered. And so you need an engineering diagram to show you properly how all of these machine parts fit together. But let me show you how it works. So this is another diagram that shows you the cross section with the microtubules up there, the 2 in the center and the 9 pairs going around the outside edge. But there are a couple of important proteins. One is called a diene. That's shown there, kind of pointed to a couple of them there for you. Those dienes reach over and grab a hold of the microtubule that's next to them and push down on it, which is very similar to how your muscles contract. You have a motor protein like the diene that reaches over and grabs on the actin fiber and is involved with muscle contraction. So something similar. So you got these little motor proteins that reach over and grab the neighboring microtubule and push down on it. But there's another important protein here. This nexin actually holds the pairs of microtubules together. And this is very important, because if the microtubules weren't held together, what would happen if that little nexin wasn't there linking them together? The diene motor proteins would cause the microtubules to slide past one another like they do during muscle contraction. That is, if they weren't connected together. This pair of microtubules, like you see on the far right, would slide down past the other one. But they're actually held together. You have these nexin linker proteins that hold the microtubules together. So instead of one sliding down past the other, they bend together because they're held together. Michael Behe describes the cilia as irreducibly complex. He says, what components are needed for a cilium to work? Ciliary motion, he says, requires the microtubules. Otherwise, there would be no strands to slide. Additionally, it requires a motor, or else the microtubules of the cilium would just lie stiff and motionless. Furthermore, it requires the linkers, the nexin linkers, to tug on neighboring strands. He's referring to the dienes there, converting the sliding motion into bending motion and preventing the structure from falling apart. All of those are required for one function, ciliary motion. You have to have all of those there, and they have to be there at the exact same time for it to function. And Darwinists have to explain how you have something like this, something that won't function without all of these parts. And why would the cell just keep making it? Generation after generation after generation. Why would it keep making those individual parts that have no purpose? Like that mousetrap. For Darwinists to explain something like that, it would have to work with just one part. So it would have to be a mousetrap, but not as good of a mousetrap with just one part. And then if you have the second part, it would still work, and it would be a slightly better mousetrap, but it would still work with just 2 parts. And then a third part, you're still going to work as a mousetrap, but a little better. It would have to work like that, but these don't. You remove any one of the parts, it will cease functioning. That's why they call it irreducible complexity. You cannot reduce it down to individual components that each serve a purpose. Let me show you another molecular machine. This one's the microtubule. Very similar to the cilia, very similar structurally as well, but it spins in a circle instead of moving back side to side. The cilia moves side to side. This is a flagellate, and it's got a long flagellum that's sticking out the end, and when it gets it out, it'll spin it. And it spins it and uses it as a motor to push itself or pull itself. Some push themselves, some pull themselves. Look at this one. This one's pulling itself with the flagellum. So you got a little, what looks like little hairs on the end that's spinning, and it spins this to pull itself through the water. So very similar to cilia, except the cilia move side to side and do so in a concerted manner. So you got a whole bunch of cilia all moving in a coordinated wave-like manner, like the waves that we do in stadiums. So they all have to move synchronously. Whereas the flagellum, it spins, and rather than moving side to side, and doesn't tend to exist in mass numbers. But both Eukaryotes, which are the kinds of cells that you're seeing here, and bacteria, bacteria are different from the other cells that make up organisms, because they're called a prokaryote. They're much smaller, and they don't have some of the organelles, but bacteria have them as well. Bacteria have flagellum. This is just an image of bacterial swimming around with their flagellum. You can see how fast they can move. I would really like to get how fast are these moving, really? I mean, let's move fast. And you got a couple stuck. You want to help that one. He's stuck on the slide, you can tell. You want to give him a little nudge, kind of help him get loose. Poof. Oh, he's like, someone help me. I'm stuck over here. Well, they move crazy fast. I would like to kind of get an idea of how fast is that? Compared to speeds, how quickly are they moving their entire length per second, how many links per second or something, and then be able to relate that to miles per hour or something. How fast are they actually moving? But really fast. And they're doing this with the flagellum. Actually, they have a series of flagellums on the end, but these all spin. Diagram of what a bacteria would look like with its flagellum there in place, if we could magnify it enough to see it. But bacteria are crazy small. You can see them under a standard compound microscope, but you have to know what to look for, because they're really, really tiny. David Rosier, who is a flagellar expert in the journal Cell, said the flagellum resembles a machine designed by humans. If it resembles a machine designed by humans, perhaps it is a machine, but designed by someone else, far superior to ourselves as well. It is the world's smallest rotary propulsion system, the flagellum is. I mean, there's no other way to describe it. It is a machine, and is a rotary machine, just like the rotary machines that we make. That's what it is. It's got a rotor and a stator. It's got a U-joint. It's got all of the parts that we would ourselves attach to a rotary, to a machine like that. And it is incredibly complex. Now remember, this is the cell that Ernest Haeckel described as a shapeless little lump of mucus or slime. Look at this machine. This is the machine right here, possessed by the bacteria. A shapeless little lump of mucus or slime. It is not. We could almost sympathetically understand why Darwin and his contemporaries could leap to such an assumption that life had evolved all on its own through purely natural processes, that the origin of the first cell was possible in that warm little pond, if it was nothing but a little lump of mucus or slime. We could almost understand that perspective. But now, knowing what we know now about cells, for evolutionists of our day to still, with great faith, believe that something as complex as the cell could just pop into existence all on its own through purely natural processes, random chance, that they still, what great faith is required to believe that something like the cell could just suddenly develop. But they try to come up with theories to explain even the most complex things. The flagellum, one of the most common explanations for how the flagellum was developed by bacteria is through what's called co-option. So what they do is they look to see if some of the parts of that machine are present elsewhere. And they say, well, they didn't really make the flagellum from scratch. All it did was take the parts from elsewhere and assemble it and repurpose it into something else. Well, for the bacterial flagellum, they usually point to this device. It's called the type 3 secretory system that is possessed by some bacteria. It does have a few of the parts that are possessed by the flagellum. You can find this in cells, and you can even find this throughout kingdoms of organisms. You can find the same part is possessed by many different organisms and or used for many different purposes in a cell. Because, I mean, that's what we do too. If you break down a motor, a big motor out of one of our engines or something, you're going to find some of those bolts or some of those washers or things that are used on many different machines. That one number 5 bolt or something, that's not the only machine it's used in. We use them in, we do the same thing. And that's how God made much of the creation as well. But what this is, this type 3 secretory system, it is actually a little harpoon that is used by bacteria to inject poisons into prey, if you will. This is actually the system, the bacteria that possessed a type 3 secretory system is the one that's actually was responsible for the bubonic plague, or the Black Death that killed so many people, this bacteria. It would just inject poison into a cell to kill it. So getting a little aggressive about its decomposition role, you know? It's like, well, you may not be dead now, but you may be, I don't know. But this is a real nasty bacteria that possessed the type 3 secretory system, used again as a little harpoon. But it does have a few of the proteins that are possessed by the flagellum. But it only, the flagellum has like 40 different structural proteins, and only 10 of them are found elsewhere in systems like the type 3 secretory system. Of the 40 structural proteins possessed by the flagellum, 30 of them, full 3/4 of them, are completely unique. Found nowhere else in any cell, in any organism. They're completely unique to the flagellum. These molecular machines are powerful arguments for intelligent design. How do you get a machine like this to just, you know, through numerous successive slight modifications, develop over time? It requires great faith. Great faith, evolutionists must have, for sure. But I think the most powerful argument for intelligent design that there is comes from the discovery of information in cells. We've discovered information inside cells. And everything we know of information says that information comes from only one place, one source, and that's an intelligent mind. Information comes from an intelligent mind, and we found information in cells. That should have led inexorably to a conclusion that the cells must have come from an intelligent mind. Because in all human experience, in our experience, information comes from only one source, and that's an intelligent mind. Powerful argument for intelligent design, the existence of information that was discovered in cells. And you probably know what information I'm talking about. If not, we're talking about DNA. And DNA is found in cells in the nucleus, the center of a cell. And there's a compartment that houses the DNA called the nucleus. And DNA typically modeled like the one there on your left in that shape of a double helix. It's kind of two strands parallel to one another that kind of twisted into a corkscrew shape. Now the double helix of DNA was actually discovered in 1953 by James Watson and Francis Crick, who won the Nobel Prize in 1962. For the discovery of DNA. I'm going to mention Francis Crick later, so wanted to bring up who he was. Now, although we tend to see the DNA looking like this, often when you see it in images, you'll see it there in the nucleus looking like the ones you see here, that kind of X-shaped structure. DNA exists in long strands in cells called chromosomes. Long strands called chromosomes. Humans have 46 chromosomes that range in size from 1.7 to 8.5 centimeters in length. And if you attached all of the chromosomes together end to end that are in just one of your cells, it would make up a piece of DNA that's at 6 foot in length. So you've got a strand of DNA 6 foot in length in the nucleus of every single one of your cells. But, and they don't usually look like this, like in this diagram. Those little X-shaped structures you see in the nucleus there are only present when the cell is in the process of dividing, because it has all of this massive amount of DNA, the 6 feet worth of DNA that's in 46 separate chromosomes, and it needs to, first it copies all of them, and then it has to separate them into 2 separate cells when it's dividing. And that would be incredibly difficult unless you had organized them in these compact structures, which is what the chromosomes you see there are. It has condensed that DNA into these X-shaped structures so it could then pull them apart without getting all tangled up. All right? So organisms have various different numbers of chromosomes, but it's not how many chromosomes you have that determine what you are or how complex you are, but it's actually what is in those chromosomes that's more important. Let me show you what I mean by this. So 78 chromosomes are possessed by dogs. Those are actually my dogs right there. That's Willie and Eli. You know, no longer with me, but I still love them. 6 chromosomes are possessed by a mosquito. 6 chromosomes. 24 chromosomes are possessed by tomatoes. Something else with 78 chromosomes that's very similar to dogs are chickens. very similar to dogs. I'm just assuming. You know, maybe dogs taste like chicken too. I don't really know. 40 chromosomes are possessed by mice. 46 chromosomes. Anyone remember what has 46 chromosomes? Humans. So 46 chromosomes. And not very similar to mice or any of these others. So it's not how many chromosomes you have, but the DNA that's on them. And let me explain to you a little bit about what this DNA is for. Okay? Now the chromosome, again, is this long strand of DNA. Again, in humans, we have chromosomes that range in size from 1.7 to 8.5 centimeters in length. And it's estimated that humans have about 30,000 genes. So the chromosome is this long strand of DNA that contains many genes. And a good analogy of what a gene is like if the chromosome is a book, the genes are the individual sentences within that book. A gene is a specific set of instructions. And estimated that humans have about 30,000 genes. 46 chromosomes means that our chromosomes contain about 1,000 genes each. And it is a little section of the DNA that contains an instruction. And the instruction that it contains, that most contain, is an instruction that tells the cell how to make proteins. It tells the cell how to make proteins. And as proteins are extremely important. Proteins are the machinery and the material that make life possible. All of those molecular machines that I showed you, those are all made out of proteins. Proteins are all the enzymes that were involved in the production of ATP, all those enzymes I was pointing out, those are all proteins that perform chemical reactions. Proteins do everything, they are everything, they are what's responsible for life. They're often also the building blocks. So for example, you have protein machines that make things like your hair and your fingernails, but your hair and your fingernails are made of pure protein. So they're the building blocks for things like hair and fingernails, and then the machines that are making your hair and your fingernails, all very important. Bill Gates said that the gene is by far the most sophisticated program around, and it truly is. It's incredibly complicated. And you have a lot of DNA, lots and lots of DNA. To give you an example of how much DNA there really is, the DNA in one human cell is about equal to 1,000 books. And that is, if you tend to see DNA abbreviated with the letters A, T, C, and G. I'll show you that here in a second. A, T, C, and G, those are the individual molecules that make up a DNA chain. If every one of those A, T, C's, and G's is equal to a letter in a book, the DNA is equal to about 1,000 books. So that's a letter-by-letter comparison of how much information that is. Or another way of looking at it, if you have a little 2-millimeter pile of DNA, a pile of DNA about the size of a pinhead is about equal to 500 stacks of books reaching the moon or a single stack of books reaching the sun. That's how much DNA there is that's how much information there is in DNA. Now let me explain what this does, what DNA does. So DNA is a code that the cell uses to make proteins. Okay, and so again, this is kind of a diagram of what DNA looks like. This is actually the RNA molecule, which is a copy of the DNA. But what the cell does is it reads this molecule three molecules at a time. So this is a long chain called nucleic acid that is made up of individual links called nucleotides. And what it does is it reads these 3 links in this chain, these 3 nucleotides, 3 at a time. And those 3 nucleotides is a code. We call them codons. It's actually a code that the cell translates. And it's a code that tells the cell how to make proteins. So the links in a DNA chain are called nucleotides. The links in a protein chain are called amino acids. So it basically reads this DNA chain and it tells the cell how to make a protein. So for example, if in the DNA you find UGC in the DNA chain, that's a code for cysteine, one of the amino acids. A CUG is a code for leucine, a different amino acid. AGU is a code for serine. It's a code that the cell translates. That's actually what they call it, translation. It translates that code into the sequence of amino acids that it uses to make a protein. most of these scientists, like Dean Kenyon and others, recognize that there's powerful evidence for design here, but we kind of have to turn a blind eye to it. Francis Crick, said, biologists must constantly keep in mind that what they see was not designed, but rather evolved. That's what you have to do. You see all this evidence of design, and you just have to keep constantly reminding yourself that although it looks like it was designed, it's not. It evolved on its own through purely natural processes, because what's the alternative? If it was designed, who designed it? God? And if there's a God, then maybe I have to think about how I'm living. And there's tremendous evidence of design. Tremendous evidence of design. Much more so than, we see by looking at something like an Arrowhead or Mount Rushmore. Tremendous evidence of design. And we just looked at a few of these highlights. If we look at cells under these high-power microscopes, or the internal architecture of the cell, all of their organelles that are there, the complex molecular machines that we've seen, like the flagellum, cilia, the ATP synthase, the existence of information in cells, tremendously powerful argument for design. Is there evidence of design? I think you have to be blind not to see it. But that's what a worldview does. You take on a philosophy, a worldview, and it kind of blinds you to what is really obvious and all around you. Tremendous evidence of design. Let me close out in a word of prayer. Father, we thank you so much that, Father, we truly thank you so much. We live in a time when we can see so much of your creation that no one ever saw before. We've seen into the internal, we see these cells no one ever saw before, in the tremendous complexity that's there, these molecular machines. And Father, thank you for making all of this, Father. And with the telescopes, we peer into the cosmos and we see the vast galaxies and nebula that no one ever saw before. Father, we're so fortunate, and we just take a moment to appreciate you for these things. We now know how vast the cosmos is, and we thank you and glorify and honor you for the tremendous work of your hands, Father. We see all the amazing complexity, and we thank you and give you glory and honor for all of this complexity. And we are humbled to know that the God that spoke the universe into existence did all of this for us, and that you love us so much that you sent your Son to die for us. Father, we're sorry that we've, even knowing this, that we sin against you, Father. Knowing that you sent your Son, that you've made this wonderful world for us, that you send your Son to die for us, that even knowing that we still sin, that we do things that we know are displeasing to you, Father. Father, I ask tonight for conviction, Father. If there's anyone with sin in their life, Father, that you would convict them. Father, convict me. Father, we ask for the renewal of conviction, a conviction for sin that maybe we once felt and became desensitized to or cold to. Father, renew that conviction. Help us to eliminate the sin in our life so that we can live the life that will honor you, Father, and have that relationship with you that we want. Father, we praise you. We glorify and honor the name of the Lord God Almighty. We praise your holy name. Thank you, Lord. Thank you for everything that you've done for us. In Christ's name, we pray all things. Amen.